Okay, so here is the metal-containing units in biology. There is something is called choice, uptake, and assembly. So inorganic elements in periodic table utilizes in biological systems such as alkaline earth metals like sodium, iron, potassium, magnesium, calcium ions required neutralize the charge of simple inorganic ions such as phosphate and sulfates, as well as complex species such as nucleotide triphosphate, DNA, and RNA. So first row transition metals ion are also important in biology, such as iron, the most abundant transition metals in Earth crust, copper, and zinc. So the bioinorganic chemistry of second row transition elements, vanadium, manganese, cobalt, so on, the only naturally occurring, also very important. Tungsten, as well, added recently to the list of metals found in biology. Here is the active site. The active centers of metalloproteins consist of kinetically level and thermodynamically stable units. Chromium 3, cobalt 3, well known in inorganic chemistry, and for their kinetic interest, interests. Okay, so the more inert second and third row transition elements play almost no role in biological chemistry. The more inert second and third row transitions elements play almost no role in bioinorganic chemistry. Technetium, platinum, and gold found place in medicine. When a metal ion is required for the given purpose, it can be concentrated by energy-driven process in a cell. This example is an uptake of vanadium into certain type of C squirt or squeezed called vanadosites. With the use of ATP requiring reaction, the element is concentrated by more than 5 orders of magnitude from the seawater into blood cells, which have almost 27 gram of vanadium per kg dry weight, more than 100 times the amount of iron. Bioavailability of metal ions. You can see this is what you have. Metal ligand here comes into the cell, passive diffusion, and then the ions will go inside from the ion specific channel. So there is the complex go inside channels of the cells, but the intake of the metal ions will go in different cells of the tissue. So now here, ions like sodium, magnesium 2, or zinc 2 solubility presents at millimolar concentration. Iron is too insoluble at neutral pH. Metal ions must be absorbed by cells to be readily available. So now tissue, pumps, cells, membrane, very important too. So everything here is talking about the uptake and a certain amount of ions or complexes. So we have some chelating agents called siderophorus that bind iron 3 tightly, which are released by bacterium into the environment. They isolate iron, solubilizing it in complex forms that taken up into the cell, where it is then released. Best studies the force in a catechol-based compound, enterobactin, and model for it is shown as well. So you can see here as well, there's a schematic showing ATP-driven uptake of ferric enterobactin into the E. coli cells of bacteria. And you can see how the cells is opened to intake something using the ATP inside the tissue membrane. Here at the table, we have the elements and the crucial average of ppm, part per million concentration. In the seawater, how much we have, the element, the crystal average in part per million as well. You see in the seawater how much we have. So now you have the enterobactin structure. This is a ligand and chemical structure of MECAM, synthetic model of enterobactin. This is look-alike this one. Enrichment strategies and intracellular chemistry of low abundance metals. So always mechanism suggested by which of a cell can take up concentrated specific metal ions from the environment, and then how can get inside the cell, vanadium, reduce, binds, and so on is being talked about it. And then for neutral metal complexes, cellular uptake can occur by passive diffusion across cell membrane. This mechanism is the one that apparently operates for anti-tumor drug cis-diamine dichloroplatinoum 2. We call it cis-DDP or cisplatin. Now, what is the cisplatin here? Sorry. Cisplatin, you can see here, is dichloride diamine platinum and in the cis structure. So cis diamine dichloroplatine administered intravenous. We call it IT, intravenous or IV. In medicine hospital, they use IV injection. So they call it IV. Injection as aqueous saline solution. Almost half of the platinum binds to serum proteins and is excreted. The rest distributed among various tissues. In blood plasma, the drug remains largely as cisplatin, owing to the high chloride ion concentration, about 0.1 molar. As a neutral molecule, cisplatin diffuses passively across cell membranes into the cytoplasm, where it's encountered a lower chloride concentration, is about 3 micromole. Hydrolysis produce cationic complexes such as cis-platin chloride hydroxyl that diffuse to DNA. So itself polyanion, where they bind to form cytotoxic lesions. The hydrolysis reaction of cisplatin is an important aspect in biological activity. The figure shows the chloride ions are displaced in a stepwise manner to form aqua and hydroxyl species These hydrolyzed forms of the drug react rapidly with the DNA than cisplatin does. What is important here is by changing its chemical form, transforming from a neutral molecule to a cation, the drug becomes trapped within the cells where it is able to damage their genomes of the cancer, binding to the DNA, and exhibit anti-tumor activity. Little toxic side effect of the drug is present on formation of cis diamine platinum by 2 DNA adducts. So here you can see the IV introducing the cisplatin into the bloodstream. Okay, so the cisplatin is like this, dichlorodiamine platin. Cis, what is it? Cis means chlor and chlor are one beside one, not opposite. So passive diffusion to the cell. This is the cytoplasm. Cell membrane. This is the cell membrane. And then the cisplatin will uptake water, release chlor, one chlorine. So the water will bind instead of one chlorine. So now introduce another water molecule, replace another chlorine. So we have two waters, two amino group with platinum with oxidation number 2, adding or removing proton hydrogen will remain OH. This is important now. It's very important. So now how many forms we have? We have one chlor, one water. We have two waters, and we have one hydroxy, one water. All these forms, active forms of cisplatin, will attach to the tumor DNA, and then the cisplatin will bind to the DNA adducts and kill the tumor. So these are, again, some types of platinum nucleobase bonds and how it is half-lives 2 or 3 hours, rate determined steps. So the original neutral molecule of the cisplatin transformed to a cation. The drug trapped within the cell and then is able to damage the genome and exhibit anti-tumor activity. So a similar process occurs for low abundant elements, vanadium and chromium. Self-assembly of metal clusters, iron sulfur cluster proteins, synthetic models. This is the redox polymer. And we have these shapes or equations, how they react. The discovery of a reaction of trichloro iron, NaHS, and this in methanol resulted cluster. And this is the cluster here, which can form or convert it to this as well. So let's see what's happening here. Protein, we have different kind of proteins, rhodopsin, and the other protein here. Cluster is 1 iron sulfur, single iron, and 4 cystoinato sulfur tetrahedral form. The formal valence will be iron 3, iron 2. This is the structure of the cluster of this protein. Iron ferredoxin, we have this type of irons, 2 irons in the oxidation state 3, and in the reduced will be 3 and 2. And so on, we have different clusters, different proteins with the presence of iron or without. Okay, now this is all introduction to the bioinorganic chemistry. What you have here is structure of S, platinus, ferrodoxine, and its 2F2S cluster. So we have ferrodoxine. You see the structure here. So there's iron with a 3 chlorine. When they can change the structure using thiol, alkyl groups, introduce more S, so the cubic here being steady of iron and sulfur, the roots to synthetic models of iron and sulfur cluster from proteins. So this is being with the methanol and produce this dimer with a bridging thiol. And if you add more, you will reach to the anion single iron. Specialized units, you have porphyrins. They are very special units. And what we have, chorines, they are as well comprised of pyrrole rings. We have metal nucleotide complexes, very important. ATPase enzymes, which catalyzes the hydrolysis of ATP. Molybdenum binding cofactors as well, important as molybdenum species. Adenosine triphosphate ATP. You can see the magnesium bound to the ATPs, and we can introduce the molybdenum cofactor here, proposed structure, and now we have here the structure of the iron molybdenum cofactor and molybdenum iron protein. So all these in biology being prepared as models as well. Control utilizing metal ion concentration in cells. So what is the beneficial and toxic effects of metal ions? As well being studied, we have a presentation of beneficial metal ions and the toxic of metal ions. It depends on the metal concentration for the cell growth. Solubilization, uptake, and transport. There is in mammals, iron absorbed from the food must be transported throughout the body. This function accomplished by proteins of the transferrin family, glycoprotein with molecular mass of about 80 kilodalton, is very big, that tightly bind to iron 3 ions. These proteins allow solubilization and transport of iron under conditions where the free ion is insoluble. So these proteins are also bind to carbonate ion and then release iron within the cells. Transferrin, like enterobactin, also binds to membrane surface receptor for uptake into the cell. So all these we are talking about are how to introduce a molecule into the bloodstream or uptake from the environment, and then the membrane of the cell will allow certain amount, certain concentration to go inside the cell, and then inside the cell it will become ionic and will be active, and how which enzymes are introduced or being responsible for such a thing. So this is the active site of structure of lactoferrin, showing the coordination of the iron and the bound carbonate carbonate ions. So this is very important functional group, COO and O. So the two oxygen of the carbonyl bounded to the iron, and the other oxygen bounded to other surrounding. Metalloregulation, uptake and storage. There is a storage as well. So a protein called FER, iron uptake regulator, which regulates a large number of genes in coordinative manner. This protein is a dimer with molecular mass of 17 kilodaltons, appears to bind specific segment of DNA. So again, these are very important for uptake and storage. And we have the mechanism how the iron ions inside the cell tissue or membrane. And then there are receptors, how they take the ions and go inside the cell, bind ATP with a proton, ADP with phosphorylate release of iron, and so on, ferritin involved, and then release through the cell membrane. So here are the actual biological molecules. Toxic metals like mercury, how mercury entered the membrane. So you can see here some bacteria uptake and transport the mercury, as well as detoxification, which has the effect of cleaning up the external environment of bacterium. This is also regulated by metal ion concentration. So the enzymes involved in the mercury detoxification, mercuric ion is highly toxic for many organisms because of high affinity attraction to thiols in variety of proteins and enzymes. Normal metabolic process can produce species like mercurials that are even more toxic than mercury to itself. So again, if there is a mercury introduced, so the cell will defend itself. If there is a mercury there, so it will bound it and then cleave it to remove the toxicity. Generation of ionic gradients. This as well I talked about. If there is a sodium intake, potassium outtake with ATP and water, so sodium will be out, potassium will be in, ATP and pi. And outside of the cell, what will happen? Scheme depicting the steps in the ATP-dependent export of 3 sodium ions and the concentrated import of 2 potassium ions by sodium plus potassium plus ATPase enzyme. Electron transfer proteins, metal binding sites in electron carrying proteins are tailored to minimize the structure of reorganizing accompanying changes in oxidation level. So there's a transfer of 1 electron, generally preferred. Coupling of proton and electron transfer efforts, a measure of redox potential control, such as iron and copper metals. So electrons can be transferred over over long distance, more than 10 angstrom. So this is electron transfer, could be transfer 1 electron or more. This is for stability. And we have seen how metal ions are taken up by cells and inserted into their sites in proteins, nucleic acids, and cofactors. We must know the specific function performed by metalloproteins, namely electron transfer, binding, and activation. Electron carriers as well. We have some biochemistry proteins have been identified measuring enzymatic activity that increases with the purification by column chromatography and fractional precipitation. So there is iron sulfur proteins having no enzymatic activity, but then detected and removed, leading to some crystalline samples of pure iron sulfur proteins. There is another type of 1 iron 0s noted before. The rhodopyroxene have active site consist of high spin iron to iron 3 ions coordinated to 4 cis-tenate sulfur atoms in almost tetrahedral shape. And so on. We discuss the 2 iron, 2 sulfur, well defined in platinuses. Ferridexine reveal the presence of this unit. And this cage, how these 3F, Fe, 4S as well. So we have many structures involved with iron and sulfur. How many of them? These are structures of clusters and crystallographic study of synthetic models. Two more highly oxidized clusters are slightly compressed along one direction, whereas the most reduced one is elongated along one direction. So again, we have these clusters, very important in biology. Cytochromes, heme contain proteins. So these proteins contains iron porphyrins as redox active centers. And cytochrome P450, like hemoglobin, have only one protein-derived ligand coordinated to the iron, leaving the open coordination site to oxygen binding. So most cytochromes have two affording coordinations, saturated octahedral complexes. These cytochromes very important. Now, if you have the iron 2, iron 3, we have 2, 4, 6, we have 5 here into the high spin. And you remember what is this? Is a tetrahedral, which is opposite to the octahedral. The octahedral will be T2G, EG. This is the ferric, this is the ferrous in different spin. And here the tuna cytochrome C from the tuna structure. Long distance electron transfer as well being found. Cytochrome C can transfer electrons far away. So iron 3, ruthenium 3 will be fast to be iron 3, ruthenium 2, and this will go to iron 2, ruthenium 3, intramolecular electron transfer. So these compounds can transfer electrons as well. Substrate binding by activating a non-redox mechanism. Hydrolytic enzymes. We talked about this box before, which is the core of the amino acids. There is a carbon, double bond, oxygen, nitrogen, amine. So it's amino acid, contain the amine and the acid. And there is a different functional group coordinated or bonded to nitrogen side or the carbonyl side. These are very important to the structure of the amino acids. And we talked about this before. Is it? So the monodentate carboxylate, bidentate. This is carbonyl. This is a bridging oxygen from the monodentate bridging. This is bidentate oxygen. Imidazolyl binding from the nitrogen side. This is the amino group to the metal. Here is the syn coordinating to the metal, and this is the anti. Carboxypeptidases, structural study as well, we talked about. And that was the carboxypeptidase A. I don't know why this is being repeated. Thank you.